Most people get infected with one or more of these viruses. It resolves on its own, with basic rest while feeling miserable. Rarely the coronaviruses that infect animals can evolve and become a new human coronavirus, which then infect and spread between people. Important examples of these types of coronavirus include the severe acute respiratory syndrome coronavirus, also known as SARS, in 2003, and the Middle East respiratory syndrome coronavirus, also known as MERS, in 2012. In December 2019, Wuhan, a city in the Hubei province in China, noticed several unusual cases of pneumonia in the hospital. These patients most notably presented with clinical symptoms of dry cough, dyspnea, fever, and bilateral lung infiltrates on imaging. The coronavirus cases were eventually reported to the World Health Organization country office in China on the 31st of December 2019. Many of the cases were reported, and searches for the source have shown that the Huanan seafood market, a wet market, as the origin. The market was where a large variety of vertebrate and invertebrate animals, wild caught and farm raised, are sold. On January 1st, said markets were closed. On January 7th, Chinese scientists identified the pathogen responsible for the unusual pneumonias as the novel coronavirus. On January 11th, China records its first death, and a day after shares the genetic sequence of the novel coronavirus with the world, which would be important in finding or establishing a diagnostic kit for the novel coronavirus. On January 13th, the first case of the novel coronavirus outside of China was detected in Thailand. On January 20th, the first United States case was reported. And this was quickly followed by the identification of the first 3 cases of the novel coronavirus in Europe on January 24th. By the end of January, the coronavirus was found to have originated from wild bats and belonged to the similar group as the severe acute respiratory syndrome coronavirus, also known as SARS. And so hence, this coronavirus is known as the SARS coronavirus 2. On January 30th, the World Health Organization declares this SARS coronavirus 2 a global public health emergency. The first death from the coronavirus outside of China was in the Philippines on February 2nd. On February 11th, the World Health Organization announced that the official name of the disease would be COVID-19, a shortened version of coronavirus disease 2019. And the cause of COVID-19 is the SARS coronavirus 2. The first death in Europe would occur in France on February 15th. On February 19th, the COVID-19 outbreak occurs in Iran. March 11th was the day COVID-19 was declared a global pandemic. It is the first pandemic caused by a coronavirus. On March 20th, Italy surpasses China as the country with the highest death toll. March 24th, Japan's Prime Minister and the International Olympic Committee agreed to postpone the Olympics until 2021 amid the outbreak. On March 26th, the number of cases globally exceeds 500,000. On March 29th, the United States has the highest number of cases globally, followed by Italy and Spain. On April 2nd, the number of cases globally surpasses 1 million. And the case continues. COVID-19 is a concern because it is highly transmissible. Preliminary reports suggest that the reproductive number, the number of people that an infected person will infect on average, is approximately 2.2. The reproductive number is basically used to reflect how infectious a disease is. The higher the number, the more infectious the disease. If we compare COVID-19 to the common cold caused by influenza, the reproductive number is about 3. For smallpox, it's about 6, and for measles, it's about 9 to 18. And so the higher, the more contagious the disease is. The number of cases of COVID-19 is increasing at a rapid rate. And this is due to the contagiousness of the virus, but also due to an increase in awareness and the availability of diagnostic kits. As of April 6, 2020, COVID-19 has affected more than 1,300,000 people and has killed over 70,000 people worldwide and carries a mortality rate of around 5.5%. But remember, the mortality rates varies between countries. Italy, for example, had a mortality rate reach up to 10%. To put this into perspective, if you compare these figures to the other coronavirus outbreaks, the SARS, for example, in 2003, had over 8,000 cases with a mortality of about 9.5%, and MERS, just over 2,500 cases with a mortality of 35%. Despite having a lower mortality rate than its predecessors, 5.5% is still very significant, especially with the rapidly growing number of cases. You can imagine for every 100 cases, around 5 people may die. If we compare these statistics to the seasonal flu caused by the influenza virus, which affects millions, the mortality rate is less than 0.2%. COVID-19 is caused by SARS coronavirus 2, a beta coronavirus, whose name derives from their characteristic crown-like appearance in electron micrographs. It is composed of a single-stranded ribonucleic acid, RNA, protected by lipid bilayer and membrane proteins, which also contain the surface proteins called spikes. The virus targets and infects the respiratory system and is transmitted by contact, droplets, and fomites from another infected person who is symptomatic or even asymptomatic. The incubation period, which is the time from infection to appearance of symptoms, is about 2 to 14 days. During the incubation period, the virus slowly triggers a response within the lungs. The lungs contain sacs of alveoli. Here we're looking at one alveolus. And the alveoli are where gas exchange takes place. Oxygen we breathe in, carbon dioxide we breathe out. The alveoli are made up of alveolar cells. One of the functions of these alveolar cells is to produce what's called surfactant, which coats the inner lining of the alveoli, which helps keep the alveoli open and allows for oxygen and carbon dioxide exchange. SARS coronavirus 2 mainly invades alveolar epithelial cells, resulting in the respiratory symptoms. Let's take a look at one of these alveolar cells in more detail and look at how the virus invades and replicates within the cell. The virus targets and binds onto ACE2, a receptor, as well as an enzyme on the surface of type 2 alveolar cells, via the surface proteins we learnt called spikes. ACE2 is needed for virus to gain entry inside the cell. ACE2 is needed for the virus to gain entry inside the cell, and this suggests that SARS coronavirus 2 may have a similar pathogenesis to SARS. And so this image will be a representation of the coronavirus life cycle within the cell. Coronaviruses generally will enter via endocytosis or direct fusion of the viral envelope with the host membrane. Once inside, the virus particle is uncoated, and its genome enters the cell cytoplasm. Since coronaviruses have a single positive stranded RNA genome, they can directly produce their proteins and new genome in the cytoplasm by attaching to the host's ribosomes. The host's ribosomes will translate the viral RNA to make proteins that will make RNA polymerase. The RNA polymerase will read the positive strand of RNA of the virus again to make a negative RNA strand. The negative strand will be used by the RNA polymerase again to make a positive RNA.
COVID-19, or coronavirus disease discovered in 2019, is responsible for a global pandemic. COVID-19 is caused by a virus called SARS-CoV-2, or Severe Acute Respiratory Syndrome Coronavirus 2, because it's genetically similar to the SARS coronavirus, which was responsible for the SARS outbreak in 2002. Now, coronaviruses that circulate among humans are typically benign, and they cause about a quarter of all common cold illnesses. In COVID-19, what happened is that there were coronaviruses initially circulating among bats, which are a natural animal reservoir that seems to have mutated and ultimately started causing disease in humans. The outbreak began in China, but has since spread around the world. As of May 1st, 2020, or roughly 5 months into the outbreak, there have been over 3,329,740 confirmed cases of COVID-19 and 237,647 deaths, resulting in a fatality rate of approximately 7%. However, current studies suggest that the actual fatality rate is likely to be lower, around 0.7%. The reason for this is that there are a lot of undiagnosed COVID-19 cases, which makes the actual number of cases go up. Many of these are asymptomatic carriers. In fact, the National Institute of Allergy and Infectious Diseases has estimated that 25 to 50% of the cases may remain asymptomatic. During this worldwide pandemic, there are many lessons to be learned from how different countries have responded to the disease. To explore that, let's use an epidemic curve which shows the number of new cases in a country seen each day. The horizontal line represents the capacity of the healthcare system within that country. Healthcare capacity accounts for things like the number of beds and ventilators, as well as the number of healthcare workers and resources like personal protective equipment, or PPE, they have available. Usually, the healthcare system is working near full capacity. So, when a pandemic like COVID-19 breaks out, even a relatively small increase in the number of patients can overwhelm the healthcare system. So, the two strategies to tackle this problem are to flatten the curve and raise the line. Flattening the curve is focused on diminishing the total number of people that get sick and slows down the rate at which new people get sick, while raising the line helps to actually increase healthcare capacity. Let's take a look at how different countries apply these two strategies, starting with China, the first country to deal with COVID-19. China informed the World Health Organization about COVID-19 on December 31st, 2019. A few weeks later, on January 24th, China aggressively tried to flatten the curve by placing a major lockdown on the Hubei province, and then issued a similar lockdown on other regions within China. Authorities forced residents to stay at home, except for essential activities like going to the pharmacy or getting groceries. Non-essential businesses and schools were shut down, public transportation was shut down, and roads between cities were blocked off. Going even further, some communities enforced a system by which there was only one entrance and exit, and everyone passing through was screened for symptoms of COVID-19. If anyone in the community tested positive, the entire community might be quarantined. In all, this affected 15 cities and about 57 million people. The measures were swiftly enacted and strictly enforced, and 12 days later, on February 5th, the exponential growth broke, and the number of new cases started to fall off. Meanwhile, given how many cases there already were at that point, China made efforts to raise the line. They built multiple hospitals dedicated to COVID 19 patients and flew in doctors and nurses from less affected regions of the country to staff these hospitals and kept them protected with well designed PPE that covered them from head to toe. By March, the economy began to return to normal, and China was seeing more cases of COVID 19 from travelers than from their citizens. Now, let's look at South Korea, which took a slightly different, yet equally effective strategy. To flatten the curve, they implemented mass testing that was free, easy, and accessible. They offered drive-through testing stations where people were tested inside their car. They also had phone booth-like testing areas where the person walks in, gets tested, and walks out. South Korea was performing 15,000 tests per day. And by April 29th, they've tested around 601,000 people out of a population of 51 million, which works out to 1 out of 85 people. Those who tested positive were either sent to a hospital if their symptoms were severe, or to a quarantine facility if the symptoms were more mild. At the same time, public health workers conducted contact tracing for every case to track down individuals that might have been exposed to the virus. Because South Korea had such
So today we are presenting COVID-19 drug targets and clinical trials presented by Dr. Parasuraman. Just a little housekeeping before we get started. Please enter your questions in the YouTube comment box. So our colleague, they are watching, and they are just like that. They will note that, and then at the end of the session, they will ask the speaker. The speaker will answer for the questions. Then after that, we will thank and we will finish the program. Okay, now without further ado, I would like to invite our beloved principal madam, Dr. Grace Ratnam, to deliver the keynote address for this webinar. Thank you.
A very good morning to one and all. On behalf of the management and staff, I welcome each of you who have gathered here for this webinar. This is a third in our series of webinars we have planned even during this time of lockdown. And I take this opportunity to wholeheartedly welcome Dr. S. Parasuraman, Associate Professor, Faculty of Pharmacy, AIMST University at Kedah, Malaysia. Thank you so much, sir, for having been here with us in spite of your busy schedule and for taking all this time and effort to apprise all those who have gathered here, including our students. He will be talking on the topic drug targets and clinical trials. As all of us in India and all around the world, we are experiencing this time of pandemic, and it is so imperative that we find the treatment or vaccines. For that we need all the clinical trials which is necessary, and we need to be in that forefront. Otherwise this will be a huge loss to humanity all over the world. The whole world is rushing towards this, including India, and we have with us Dr. Parasuraman, who will apprise us of what is happening all around the world. So once again, I thank him and I thank each of you. And at this time, I take this opportunity to thank our Department of Pharmacy Practice, Each and every one of them, including Dr. Krishna Kumar, our HOD, have worked tirelessly to bring this to the forefront using this technology and using our guest speaker. So welcome each of you. Thank you.
Thank you, ma'am, for giving introduction and the keynote address. And I take pleasure in introducing Dr. Parsuraman. Dr. Parsuraman is an associate professor and unit head, pharmacology, faculty of pharmacy, Asian Institute of Medicine, Science and Technology from Malaysia. He has undergone his undergraduation and post-graduation from the Dr. Tamil Nadu Dr. MGR Medical University, Chennai. He has done his PhD from Jipmer, Pondicherry. He had worked as clinical research associate for Trident Life Sciences. To his credit, he has published more than 70 papers in various national and international journals. He is currently working on industrial and petrochemical toxins, which has a greater impact on global environmental safety. At this moment, I take pride in inviting Dr. Parsuraman for the webinar organized by C.L. Bade Mehta College of Pharmacy. Thank you for joining with us, sir. Over to you now.
Good morning, madam. Thank you for your brief introduction. First of all, I would like to thank the management of C.L. Bade Mehta College of Pharmacy and the Department of Pharmacy Practice and my close friend Dr. Krishna Kumar. So all the times I used to get communicated with him to have update what's happening around Chennai. He used to give insights about SARS, no more COVID, because last May 4th, US government as well as US FDA changed name as a SARS-CoV-2, severe acute respiratory syndrome coronavirus. That is abbreviation, severe acute respiratory syndrome coronavirus 2, no more COVID-19. Okay. So that many of us, we are habituated to say name as a COVID-19, but no more COVID-19. Since May 4th, SARS-CoV-2, SARS-CoV-2. So today we will share our knowledge on the available targets and clinical trials, what's going around the world. So I will take you into the subjective matter, so which may be helpful for future generation as well as for the pharmacist. So again, no more COVID-19, severe acute respiratory syndrome coronavirus 2. All the public forum, the name was updated. Okay. So that we'll take note on this, and also this coronavirus binding into the human cell through a spike protein. That is very, very linking point where the virus as well as human cell is getting connected. So the target is main target is spike protein, otherwise called it as S protein. S protein is playing major role to spreading the disease. First, in the 2019, end of the year, December, many people in China, they got admitted in hospital with respiratory syndrome. That time, the consultant physician, Li Wenlian, he was worrying about why many people are getting and coming and admitted in hospital, and also the spreading rate is very, very high. Finally, he suspected that this is similar to the SARS, which was happened in 2006, severe acute respiratory syndrome. The symptoms, everything similar, and the spreading pattern also similar, and he reported into the hospitals. But around the scientific community and committees, they are not accepting this is actually communal disease. Later on, the outbreak boomed in the year 2020 in the month of January, first week onwards, and many people start dying with particular disease. Finally, they found that this is actually similar like SARS, but through coronavirus 2, not similar that earlier coronavirus. So coronavirus 2. So that time they named as a COVID-19. COVID-19. So first of all, many of us we are not clear about virus and the other pathogens. Other pathogens like bacteria and fungal, they are having the defined cell wall, or some of the pathogen they are having the cell membrane, but virus not having both. Because some of the antibiotic and pharmacy, when you're reading the pharmacology and pharmacotherapy, we'll read about cell wall synthesis inhibitor or cell membrane inhibitors. So cell wall synthesis inhibitor mainly for antibacterial function, cell membrane inhibitor mainly for antifungal functions. So that, but virus not having both cell wall and the cell membrane. So that is very important place. So we cannot have any drug which is going to inhibit cell wall synthesis or cell membrane synthesis. So that we have to go for alternative ways. So mainly the virus having the integrated protein on the outer shell and also some constructed with membrane protein as well as spike protein, both membrane protein as well as spike protein. And also one more important thing, bacteria having its own metabolic process, but whereas virus not having or do not carry out any metabolic processes. So these are the two major difference between virus and bacteria. Why? Because bacteria will have defined cell wall and also it will carry out its own metabolic process. But whereas virus not having cell wall or any cell membranes and also it do not carry out any metabolic process. So these two are the major difference between bacteria and virus. Then how question is how virus is multiplying. Virus is much depending on to the host metabolic pathway. That is very very important place where virus is getting multiplied or rapidly spreading. So usually virus will insert its metabolic DNA or RNA material into the host cell. So host cell will replicate, then finally host cell will get rupture and the virus will get or daughter virus will get mixed into the bloodstream or it will be coming out and spreading later. So usually virus are different types in genome. They are actually DNA virus, RNA virus or RNP viruses. So DNA virus, it will be herpes and hepatitis B or DNA virus, some of the examples. What is RNP? Some of the viruses call it as RNP virus, ribonucleoprotein virus. Mainly influenza will come under RNP virus called ribonucleoprotein virus. But whereas RNA virus are very dangerous. Hepatitis A, hepatitis C, hepatitis D, hepatitis E, retrovirus, it will be include HIV virus, then coronavirus. All these are RNA virus. We can have the drug target for DNA virus and RNP virus. Why? Because DNA always it will be stable structure. RNP also protein actually. But thing is RNA keep on will change the structure so that we cannot have any target on metabolic pathways. We can go with either with interlinking proteins or it will be nucleic acid, some other nucleic acid or it will be byproduct like that. So RNA virus is very major problem for developing drug for RNA viruses. So that only we are not having any particular drug for hepatitis A, hepatitis B, hepatitis E, and also retrovirus and coronavirus. We are having only some minimal drug targets, but we are not effective drug targets. We are not having for RNA viruses. So how it was get spread that actually? So severe acute respiratory syndrome coronavirus 2 SARS-2 is a positive science single standard RNA virus. So the virus will have single standard RNA virus. It was believed that this is spread from bats actually. So most of the RNA virus, whatever we are having RNA virus, most of the virus is spread from bats. So DNA virus not a major issue, but RNA virus is a major issue in the society. The spreading also very rapid compared to DNA viruses. So what is a coronum? Coronum means it will be in Latin because in this, if you see the cell structure around the cell membrane, you will have the spike protein. will be like crown. Many of them, it will be, this is actually S protein. Whatever is the crown, above that S protein, in between blue color is actually membrane protein, and also envelope protein. The three are the integrating the virus actually. Spike protein, envelope protein, membrane protein, or membrane glycoprotein. Inside that, the virus will have RNA. So that whenever the virus is getting entering into the host, the spike protein is playing very major role. Why? Because the spike protein will get intact with the host cell, thereby it will be making the small foreign structure on the host cell and inserting its genetic material, that is RNA, into the host cell, so that the host cell now it will carry the RNA from the virus. Then host cell will start work for this virus, then thereby replicating, and it will be finally virus is multiplying rapidly. So that this particular coronavirus having the 4 different genera. So alpha coronavirus, beta coronavirus, delta coronavirus, gamma coronavirus. So 4 genera of the CoV virus, alpha, beta, delta, gamma virus. So that we are not having major problem with alpha, beta and gamma viruses. All the previous history, MERS, Middle East Respiratory Syndrome coronavirus, also beta coronavirus, SARS, Severe Acute Respiratory Syndrome coronavirus, that also beta virus, SARS, COVID-2, Severe Acute Respiratory Syndrome coronavirus 2, this also, all these 3 are beta virus. So indicating that alpha, delta, gamma coronavirus is not a major issue currently, but beta virus is a major issue. So all these 3 viruses believed that spread from bats. So in some point of time, the MERS is a major problem, that is mainly in Middle East. Middle East means Saudi Arabia region. Then SARS in 2006 and 2007, mainly in Southeast Asia. So now SARS-CoV-2, Severe Acute Respiratory Syndrome coronavirus 2, throughout the world. So all the world we are facing problem with now this beta coronavirus. SARS-CoV-2 beta coronavirus, we are facing major problem. So how it's getting spreaded. So in this coronavirus also there is 2 different types: low pathogenic human coronavirus, moderate to high pathogenic coronavirus. Actually, in that HCoV 229E, HCoV NL63, HCoV HKU1, HCoV 0C43. These are the viruses getting spread from the animals, especially camel, kettles, rodents, bats, and also getting transmitted between the human being. But there is no major pandemic or major death due to these viruses. So that these viruses are classified as a low pathogenic human coronavirus. But whereas SARS-CoV, SARS-CoV-2, MERS-CoV, these are the high pathogenic because the incident of mortality is minimum 5% total outbreak. Minimum 5% in total outbreak. SARS is actually from birds to rodent to human being, then transmitting in between humans. MERS also from birds to kettles. Okay, but we are not having clear evidence. MERS to kettles to human being to human being. SARS-CoV-2, bats, it will believe that bats, it will be in between, intermediate, we are not sure, directly to the human being. So human being to human being, causing the major problem. So the spreading rate also very high, and also this is getting spread through face-to-face contact. Okay. This SARS-CoV-2 direct spreading through direct transmission by face-to-face or hand-to-face contact from and contact from infected surfaces. Usually, this virus will in copper it will live for 4 hours, in cardboard 24 hours, plastic 2 to 3 days, in air 3 hours. In this also always all the regulatory agencies and government saying that they keep social distancing. But social distancing is not a method of treatment, only method of prevention. Why? Because the large particle from the patient it will be spread up to 3 feet. The smaller particle it will be spread up to 6 feet. So that only all the government is restricting and suggesting to maintain the social distance at least 3 feet between. So that is actually our hand gap, actually our hand gap 3 feet. So we can minimize the spreading rate. So and also recent reports saying that oral transmission also one of the method of or modality of spreading the disease. Why? Because fecal matter also containing this SARS-CoV RNA virus. But thing is actually clinically not described so far, because fecal oral transmission also of transmitting this virus. Why? Because the fecal sample, when I was checking the fecal sample from the infected patient, the fecal matter having the RNA from the SARS-CoV-2. So this also, but scientifically still under investigation. Then vertical transmission, mother to child also not much reported. There are few reports, but not much reported. Not much reported. So clinically, so incubation period normally 14 days, but median time of incubation period 4 to 5 days. So normally patient will have experience with fever about 80%, cough about 60%, fatigue about 44%, anorexia about 40%, shortness of the breath about 30%, sputum about 20 to 30%, myalgia about 10%. Rarely patient will have diarrhea and nausea period to development of fever. This also very rare, but most of the patients reported before developing fever, they are having the GAT problems, nausea and diarrhea. So commonly the patient used to report about fever, dry cough, flu-like syndrome. In severe cases, they are reporting dyspnea and little bit chest pain, hematosis, then respiratory insufficiencies. Complication mainly pneumonia, respiratory, upper respiratory tract infections, and the liver dysfunctions, arrhythmia, cardiac injuries. These are the complications they reported so far. Okay. In clinically, about 80% of SARS-CoV-2 patients, they are mild and asymptomatic. So that only now recently in Chennai, whenever you are checking the patient, many people when you are checking the more number of clinical interventions, increasing the number of cases. Most of the cases are asymptomatic. But recently WHO said asymptomatic type, the spreading rate is comparatively less than symptomatic one. Asymptomatic, the spreading rate is comparatively less than symptomatic one, but not well documented. So 80% of infections are mild or asymptomatic. This one not required hospital administration. This one not required hospital admission. About 50% of infections are severe, but these cases require oxygen therapy. About 5% of total infections are a little bit critical, and this patient require ICU interventions, ICU admissions. So atypical about usually they are having the delayed presentation of fever and respiratory syndrome, and in the asymptomatic, about 13% of the children they stay. They are 13% of children they are asymptomatic ones. So how we can detect actually? Many of the people in society they are assuming that the only test available for testing the SARS-CoV-2 is RT-PCR. Not only RT-PCR, even blood test will it will give the clear indications whether patient having the SARS-CoV infection or not, and also radiographic findings. RT-PCR, not only RT-PCR, blood test and radiographic findings can be. RT-PCR, many of us we know RT-PCR because all the news media every day they are telling RT-PCR test, RT-PCR test, RT-PCR test. So apart from that, even blood test scan and the radiographic intervention scan. RT-PCR usually the patient sample will be taken from patient nasopharyngeal cavities and using that sample, sputum sample, RNA will be isolated and it will be sequence will be read for COVID-19, SARS-CoV-2. So if sequence matches, or it will be by that we can interpret whether patient having the infection or not. Apart from that, in blood test, most commonly the patient, those are infected with SARS-CoV-2, they will have leukopenia, so increase in lymphocyte count. They will have lymphopenia, increase in lymphocyte count, and also neutrophilia. They will have these both are most common thing. Lymphopenia and neutrophilia. Neutrophilia means actually neutrophil will take the leukocytes and also increase in number of neutrophils in blood. Okay, call it as neutrophilia. So these both are most common thing, lymphopenia and neutrophilia. Lymphopenia is actually increase in number of lymphocytes. Neutrophilia is increase in number of neutrophil cells. And also the patient will show increase liver enzymes, serum ALT level, AST levels. And also patient is to show elevated lactate dehydrogenase enzyme, and also patient is to show increased level of interleukin 6 and IC reactive protein CRP. IC reactive proteins, increasing level of interleukins and IC reactive proteins. These are the levels indicating that the elevation of these are the levels indicating that patients are having the SARS-CoV-2, and also patient is to have high fibrin levels. So these are the things indicating that the presence of the virus. The CRP protein actually one of the increasing in the response to inflammation. This is very much commonly observed in severe cases, and also high ferritin level. Usually the protein that stored iron and release it in the controlled manner. So usually the patient will have more iron content in their blood. So these are the things indicating that one is lymphopenia, neutrophilia, elevated liver enzyme level, elevated antioxidant level, LHD, LDH, lactate dehydrogenase, increase in interleukin levels, and increase in level of C-reactive protein, and increase in level of ferritin. This indicating that patient they are having they are infected with SARS-CoV-2. In radiological finding, CT scan and chest X-ray will demonstrate presence of biliary aspase, consolidation. So this will be indicating that same like X-ray is commonly used for checking the tuberculosis. Same like X-ray also can be used for to determine to check presence of SARS-CoV-2 or not. So apart from RT-PCR, blood test can be done and the radiological interventions can be done to determine because sometimes what will happen, waiting for the RT-PCR, sometimes you may not get the reagent and sometimes the test cost may be get increased so that we can go for alternatively blood test and X-ray or CT interventions. Okay, so now the disease having the three different types actually, mild to moderate and severe disease and hypercoagulatory condition. So these three conditions usually. So mild to moderate disease, patient may not require any hospital, some may not require hospital administration, not compulsory. Treatment is based on the case by case basis. They require supportive care and ability to patient self-isolation at home. Okay, mild to moderate condition. Okay, supportive care and the ability to patient should have ability to take care themselves and also they are having provision to get isolated from their family. Okay, so this is actually method of management, mild to moderate. Many of the people they used to follow this one when they are having any known symptoms, fever, sore throat, and also it will be tiredness, fatigue. Automatically they will go for the self-isolation. Okay. So severe condition, they need hospital administration. Okay. And also patient will have pneumonia, respiratory failure, sepsis, shock, cardiac myopathy, and arrhythmia. Arrhythmia may cause acute injuries, kidney injuries will occur. And also in prolonged hospitalization, patient may get secondary bacterial infections because of these are the things due to the prolonged hospital administration, secondary bacterial infection, thromboembolism, and gastrointestinal bleeding, and critical illness like myopathy. These are things due to the hospital administration. Usually in severe condition, patient will have pneumonia and respiratory failure and the cardiovascular events, including arrhythmiasis and renal injuries. Okay. Then what about third one? This one is many of us we are not clear about hypercoagulatory and SARS-CoV-2. This is actually whenever many of the patients autopsy shows that there are the coagulation of the blood inside actually. In lungs, everything, the blood coagulation they observed. So that only they found that the hypercoagulability, one of the conditions with the SARS-CoV, but the reason is remains unclear. So far, we are not having any clear reason why the patient they are getting hypercoagulability. So this is one of the conditions. So one is mild to moderate, severe, and third one is hypercoagulability. Okay. Now coming to the structure, because if you know the structure, then we can very easy to discuss about targets. Okay, very easy to discuss about target. If you see the coronavirus, the above the coron, there are virus having the S protein, spike protein, then membrane protein, then envelope protein, E protein. These three are the actually covering the virus. S protein, M protein, E protein. But M protein and E protein not having major role. Only this spike protein having a major role, because this S protein, spike protein helping virus to get bind into the host cell. So virus is replicating mainly binding with host cell through the S protein. So the main target is S protein. So S protein is one of the main target. Again, one more target from host cell, that is angiotensin converting enzyme 2. Angiotensin converting enzyme 2. So we'll discuss about angiotensin later. First we'll discuss about S protein. And also this one, the SARS-CoV as having the two genotypes. So many of us, some regions, if for our India, some regions we are observing more number of mortality. Some region the mortality is very less with the SARS-CoV-2. So it will be indicating that why. Some places there are more number of incidents, some places very less number of incidents with mortality rate. So what is the reason? Because the SARS-CoV-2 having two genotypes, L type and S type. L type is very aggressive one, and also more prevalent. Approximately around 70% of the cases in the early outbreak falls under this particular type, L type. So S type is laboratory little bit older, less common type and less aggravating. Around 30% of the cases are S type. So in pandemic condition, wherever the spread of L type, more number of mortality was observed. Wherever the spread of S type, less number of mortality was observed. Then if you see the throughout the global history, some countries like USA and the United Kingdom, the mortality rate is very, very high. So some countries the mortality rate is very less. Russia, India, Singapore, there and all mortality rate is very, very less. Maybe due to the genotype. So the more number of mortality was observed in the spread with the type coronavirus. Less number of mortality observed with the spread with the S type of coronavirus. So target now, the coronavirus mainly the binding to the host cell by binding its S protein to the receptor protein. That what is the receptor protein here? The angiotensin converting enzyme 2, ACE2. So the now problem is actually S protein and angiotensin converting enzyme 2. These two are the major problem now, because the virus having S protein getting interacted with angiotensin converting enzyme 2. So now target is actually we have to destroy the S protein, or we have to increase the upregulation of angiotensin converting enzyme. Why? Because this S protein make sure that it will down regulate the angiotensin containing enzyme. So when angiotensin containing enzyme getting down regulated, automatically the cardiovascular functions also will get affected. So that only the patient, those are having known cardiovascular incident, they are having higher incidence or higher number of mortality with SARS-CoV-2. So angiotensin is playing major role. Okay. So we can ask question now. Okay, angiotensin converting enzyme is one of the enzymes playing major role in regulating blood pressure. Yes, that is different from this one, because that is very general one, angiotensin converting enzyme ACE. But this one is angiotensin converting enzyme 2. This will be this particular receptor protein present in all the epithelial cells, mainly in smooth muscle cells, intestine, kidney, blood vessels. These places, smooth muscles, this receptor protein is very, very abundant, and also type 2 alveolar cells of lungs, this receptor protein is very abundant. So that only the many of the patients they are having the okay, problem with lungs. Wherever the angiotensin converting enzyme 2 proteins are there, that particular organ is getting affected. So that only the SARS-CoV-2 patient, they complaining about respiratory failure, kidney problem, and the cardiovascular problems. And one more membrane protein, transmembrane protease serine 2. Okay, this also playing very important role, and also helping this membrane protein helping virus to get binding into the host cell by fusion mechanism. Okay, by cell fusion, so that once it will be virus is getting fused with the host cell, virus will easily transmit its genetic material into the host cell. So now one is angiotensin converting enzyme 2, other one is transmembrane protease serine 2. So that many of the world countries working on this transmembrane serine 2 inhibitor. Okay, if transmembrane serine 2 inhibitors developed, this may get this mechanism may helpful to break this fusion mechanism, and also angiotensin converting enzyme 2 inhibitor get development that will be helpful to break S protein binding. S protein binding. So now in this structure, the virus is coming, so it will be getting interacted with the angiotensin converting enzyme 2, and also so this transmembrane protease serine 2 is helping to get binded. So next slide, if you see, so once it will be spike protein is binding into the ACE2 receptor, so automatically start cleaving this one. Okay, start destroying this receptor. So by the time this transmembrane protease serine 2 is making the open, making the open, when getting opened, easily virus is getting entered inside the host cell, so that virus will replicate now easily. So that now this virus is making sure that down regulation of the ACE2 receptor, and also inhibiting the and also getting interacted with the transmembrane protease serine 2. So that mainly this one is actually making reduction of availability of cell surface ACE2 receptor. So that negative regulations. So that automatically what will happen? Automatically patient cardiovascular function will get inhibited. So again, one more thing, when ACE2 is getting inhibited, ACE2 when goes for the down regulation, automatically ADME, okay, that is actually ADME, metalloprotease domain 17, ADAM, metalloprotein domain 17 gene expression also will get affected. This will lead to the release of tumor necrosis factor alpha, TNF alpha, and cytokine such as interleukin 4 and interferon Y. So it will increase when ACE2 is getting inhibited. Automatically gene expression for ADAM, metalloprotein domain 17 also get affected. This one otherwise called as tumor necrosis factor alpha converting enzyme. This ADAM 17 is otherwise called as tumor necrosis factor alpha converting enzyme. This also will get affected, thereby abundant or more release of tumor necrosis factor, that is TNF alpha and interleukin 4, interferon alpha, interferon gamma is getting released. So this is getting this one when increasing cytokine, this cytokines concentration, automatically patient will have cytokine storm. So now all the places, many big political leader also stating that chloroquine, hydroxychloroquine. So this hydroxychloroquine and chloroquine mainly play a role on this particular pathway, pro-inflammatory pathways, and also preventing cytokine storm. So the mechanism of hydroxychloroquine and chloroquine, this both the drug prevent cytokine storm, thereby the blocking the pro-inflammatory pathways, so that thereby preventing the worst scenario of SARS-CoV-2. This is actually mechanism. So now thing is actually how we can target now. One is main thing is improve our immunity. Immunity is playing very major role for any diseases. Even cancer also, there are many scientists stating that our own immunity, our passive immunity is playing major role to prevent cancer. cancer is one of the horrible disease having a major death throughout the world. So immunity is very, very, very important. So that internal immunity system response is very important. So that we have to try to boost our immunity. That is only way now we can prevent the disease spread. And also blocking the signaling pathways, mainly signaling pathways already we seen. One is ACE2, other one is transmembrane protease arraign 2. These two are the major target. So when blocking these two targets, automatically the incident of or the spread of the disease may be minimized. So what is difference between SARS-CoV and the SARS-CoV-2? SARS-CoV-2 mainly will be bind to angiotensin converting enzyme 2 and other one Middle East Opioid syndrome. That virus will mainly bind to the DPP4 receptor. So this major difference between the two viruses. Both are coronavirus, but both will have different targets. Now thing is, one is immunity, one is blocking. So now coming to the pharmacotherapeutic agent, that is antiviral drug. Because antiviral drug play major role to minimize or prevent or inhibit the further progression of the disease. One of the promising drug is remdesivir. That is actually from beginning of the disease pandemic, the remdesivir is commonly used. Remdesivir commonly used in combination with hydroxychloroquine and corticosteroids, prednisolone, hydroxychloroquine, remdesivir. These three combinations most commonly used in many of the world countries. So recently, yes, 1 week before, Russia is approved use of avifovir for the management of SARS-CoV-2 because they tested throughout the Russia, they tested this drug and they found that avifovir is much beneficial and also helping to the reduce mortality rate. This avifovir actually generic version of Japanese flu drug avifovir. And now after seeing this avifovir drug, now Japan is taking initiation to clinical trial on avifovir. Now one is promising agent. Now clinically promising agent remdesivir. Remdesivir are commonly used for managing disease in many of the world countries. In now last week avifovir is approved by the Russia. Then after seeing this avifovir is beneficial, now Japan is initiated with avifovir with other trials. Apart from this antiviral drug, chloroquine and comostat. Comostat is actually serine inhibitor, transmembrane protease serine inhibitor, serine 2 inhibitor composite. Otherwise, POI305. Then conversion plasma and vaccine. These are the other pharmacotherapeutic agents. Now, apart from remdesivir, interferon, lopinavir, letinavir, and darunavir all shown effect in vitro conditions. But remdesivir is actually most commonly used for managing the RNA virus infections, including SARS and MERS. And US government now third trial and phase 3 trial is under investigation. So this way the antiviral drugs are helping to prevent. So coming to the remdesivir, in vitro trials shown beneficial in the month of February. It was published in Cell Research in month of February. Later on, same time, National Institute of Health clinical trial showed beneficial effect of remdesivir against COVID-19. Then United Kingdom approved this drug for the management of coronavirus infection in the month of May 26th. That is hardly 2 weeks before. So in United States, in the month of beginning of May 1st onwards, this drug was used for the managing the COVID-19, remdesivir. So remdesivir showing beneficial effect many of the conditions, so that most of the cases this drug is commonly used. So recently, 1 week before, Russia is approved use of avifovir for the management of coronavirus. And after seeing that report, WHO is now keen to see result of trials of avifovir for the management of COVID-19. This was posted 1 week before. 1 week before. 1 week before. Now, apart from that antiviral drug, chloroquine. What is the use of chloroquine or hydroxychloroquine? This drug mainly blocks the virus, by preventing the cell fusion. And also chloroquine may effective in preventing SARS-CoV-2 cellular entries. Then again, other drug, Camostat. This is actually serine protease inhibitor. Will inhibit the this transmembrane protein. So transmembrane protein, thereby preventing the entry of virus into the host cell. Then what is that convalescent plasma? Actually, the blood sample will be collected from infected patient. From that, their plasma will be isolated. That plasma will be injected to the normal human being to develop passive immunity against SARS-CoV-2. Passive immunity against SARS-CoV-2. Then finally, vaccines. But some of the vaccine trial already begins, but many trials ended with failure. Failed result. So mainly these clinical trials now on the way with the messenger RNA based SARS-CoV-2 vaccine. So how it will be? Normally, the SARS-CoV-2 will come to the cell surface. On cell surface, cell will have ACE2 and a transmembrane protease serine-2 receptor. Both it will bind, then it will be entered into the cells. Once enters, it will release its RNA. This RNA will be get binded into the ribosome of host. So translation, then proteolysis under it will go. Then through proteolysis, it will get multiplied. Same time, this other cellular part will be multiplied on the host cell. Finally, packing will occur. Once packing will occur, it will be getting out from the host cell. So by this, it will be multiplying inside the host. So that one is actually monoclonal antibodies. So now currently we are not having any effective monoclonal antibodies. So to prevent this binding site and the serine-2 inhibitor. So this serine-2 inhibitor blocked means automatically the virus cannot able to enter inside the cells. Then this one is actually fusion inhibitor, chloroquine or hydroxychloroquine fusion inhibitor. Then it will be proteolysis, lefnavir and ritonavir. So remdesivir, where it will acting, it will be acting on RNA dependent and RNA polymerase. There it will be this drug is preventing replication of viral RNAs. So by this drug preventing the multiplication of the SARS-CoV-2 virus. So now, hydroxychloroquine will come to the story of hydroxychloroquine. So during the SARS-CoV, so there will be macrophage cells. The macrophage cells will be releasing more of the cytokines. These cytokines will be produce the cytokine storm. So thereby causing the acute respiratory distress syndrome. So that in SARS-CoV-2 infection will be 3 stages: stage 1, stage 2, stage 3. Stage 1 is asymptomatic, stage 2 is non-severe symptomatic, stage 3 is severe respiratory inflammation. So now in this stage 3 and stage 2 condition, the hydroxychloroquine will be used for the managing the disease in combination with remdesivir and prednisolone. So remdesivir going to inhibit the RNA polymerase. This hydroxychloroquine going to inhibit cell fusion and also prevent the cytokines or hypercytokinemia. So thereby inhibiting cytokines, thereby preventing the acute respiratory distress syndrome. So mainly this hydroxychloroquine and cytokines are preventing cytokine storm. So now we can ask question that so there already this virus is targeting ACE receptor. Why we can use ACE inhibitors also for the managing But AC inhibitors are different from angiotensin converting enzyme 2 because this was a hypothesis, but there is no evidence that okay, we can use any of the AC inhibitors for the managing COVID. Cannot because so far in clinically no trial showed a beneficial effect of angiotensin receptor inhibitors like captopril, enlopril. No drug shown any beneficial. No trial was initiated and no drug having the shown in in vitro condition. No drug shown beneficial effect against SARS-CoV-2. So that only this is under interventions. Don't try to compare both AC inhibitor and AC2 inhibitors. Why? Because genetically both are differ. Angiotensin converting enzyme, the enzyme coding with chromosome 17, but angiotensin converting enzyme 2, enzyme coding with chromosome X. Both are differ actually. We cannot able to interlink both angiotensin converting enzyme and angiotensin converting enzyme 2. And also angiotensin converting enzyme help to convert angiotensin 1 to angiotensin 2, thereby causing the vasoconstriction. But the angiotensin converting enzyme 2 directly led to reducing the amount of angiotensin 2. Both are different mechanisms and also gene coding also differ. One is AC is chromosome 17, AC2 is chromosome X. Both are differ. So that now we are clear that the target is actually angiotensin converting enzyme 2 and transmembrane protease serine 2 receptor. These two are the targets. So all the trials based on this one. Now many of the trials going with fusion inhibitors. Fusion inhibitor means hydroxychloroquine. So far totally 778 trials are registered with the US FDA with many antiviral drugs and antimalarial drugs. It will be hydroxychloroquine. So far trials are registered with lopinavir, ritonavir, ribavirin, interferon beta, lopinavir, then it will be interferon beta 1b, ozone therapy, enrichment, enriched platelet inhibitors, prednisolone, methylprednisolone with other antiviral drugs, chloroquines, then vitamins, then hyperimmunoplasma, interferon beta 1a, then some of the studies with vaccines. So various trials are registered with this of the agent, and also some of the trials registered with nitric oxide gas inhibitor, and finally the trials are withdrawn by the particular agencies. So trials are also started begin with nitric oxide gas inhibition, but the trials are withdrawn. This may be one of the mechanisms for playing with secondary messenger levels. So some trials is hydroxychloroquine also withdrawn, and plasma therapy trials also withdrawn. So totally 15 trials are completed, 15 trials are withdrawn. Okay. But many of the trials so far research was not published. Okay. So in that some trials are suspended in between. 3 trials with hydroxychloroquine already suspended, and about some of the trials it will be in next trial, phase 1 to phase 2, phase 2 to phase 3 like that. So totally now 408 trials recruiting patients for the new drug interventions. So with hydroxychloroquine, around 40 trials are registered. In the 25 trials are recruiting subject. In that 7 trials are conducted. 7 trials in USA, 1 trial in UK, 3 trials in Canada, and 3 trials in Brazil, 2 trials in China. In India, so far it is hydroxychloroquine, not promising trials. So now some of the agent went to the phase 3 clinical trials. So total 67 trials are in phase 3. One is actually unizumab, that is actually monoclonal antibiotic. Then hydroxychloroquine, around 14 trials are in phase 3 trials. Some in Egypt, they are trying with to improve the immunity by giving the natural agents, natural agent like honey, Nagella sativa, plant extract. In this particular trial in Egypt, then brozimab, this is actually monoclonal antibiotic. Now currently we are using for managing the cancer treatment. So this drug is actually VEGF A inhibitor, then tocilizumab, then azithromycin. So in many of the trials, many of the clinical conditions, azithromycin showed beneficial effect. So that azithromycin also added along with hydroxychloroquine and also with remdesivir. So mainly azithromycin, prednisolone, hydroxychloroquine, remdesivir. Four drugs are promising agents commonly used for managing the SARS-CoV-2. So apart from these drugs, other drugs also under the investigations like trypsin inhibitor, then polyoxidine, then lefnovir, chloroquine combinations, glucosamine combinations, anti-interleukins, clarithromycin. Many drugs are under investigations. One of the promising trial is ACT COVID-19. This trial is in phase 3 in USA. This one is actually randomized trial and open label and parallel group randomized controlled trial and masking none actually, patient no open label trial. This trial is conducted with hydroxychloroquine with 1,500 participants. The trial is maybe they are expecting very beneficial effect from this particular trial. And also this trial they are using 3 drug regimens. One is azithromycin, one other regimen is hydroxychloroquine and chloroquine. Other drug is interferon beta. The 3 drugs they are using for the managing the SARS-CoV-2. Actually, in this particular trial, how they are doing is for outpatients, mild to moderate condition, they are giving hydroxychloroquine 400 milligram 2 times a daily for 7 days, or chloroquine 500 milligram 2 times a day for 7 days, along with azithromycin 500 milligram for day 1, followed by 250 milligram for 4 days. This combination they are giving, and in phase 2 and phase 1 trial they observed beneficial effect, so that they are going with this particular combination at COVID-19 trial. So combination is actually hydroxychloroquine or chloroquine with azithromycin. So in severe condition, they are recommending hydroxychloroquine or chloroquine with azithromycin and also interferon beta. So patient will receive hydroxychloroquine loading dose followed by 400 milligram 2 times a day for 7 days, or chloroquine loading dose followed by the 500 milligram 2 times a day for 7 days, along with azithromycin 500 milligram from day 1 followed by 250 milligram for daily for 4 days. Along with patient will receive interferon beta subcutaneous injection on day 1, day 3, day 5, day 7. So in phase 2 trial, they observed some of the beneficial effect with this particular combination. So that phase 3 trial was begun with 1,500 patients. So expecting this trial may give some fruitful outcome. So these are the trials completed throughout the world with hydroxychloroquine, antiviral drugs, ritonavir and remdesivir. So some of the trials listed here. Okay. So what about in India? India also will be fighting against and also involving in COVID research. And also in India so far, 11 trials are listed in India, mainly from Max Super Specialty Hospital and PGI Chandigarh and Maulana Azad Medical College, New Delhi. These three institute registered trial in US FDA website. If you are searching in our other database, we may have more number of trials in India. But currently in US FDA site, we are having the 11 trials. India also will be contributing more for discovery of pharmaceutical agents against management of SARS-CoV-2. So now recommended, this is actually recommended by Infection Disease Society of American guidelines. So they are recommendation 1 and 2, hydroxychloroquine with azithromycin. Recommendation 3 is leftovir and ritonavir. Recommendation 4, steroid. Recommendation 6, monoclonal antibodies. Recommendation 7, plasma transplantation. So these three are the recommendations given by the Infectious Disease Society of American guidelines. So, so far we discussed about many aspects of drug targets as well as SARS-CoV-2 binding sites. SARS-CoV-2 now we understood that mainly targeting on ACE2 and the transmembrane protease serine2. These are the main two targets. So either we have to target on drug for inhibiting spike protein into the angiotensin converting enzyme 2 or transmembrane protease serine 2. These two are the main bases. Apart from that, fusion inhibitor. So fusion inhibitor, that is hydroxychloroquine, also beneficial, and mainly patients are dying with other complications because when this angiotensin 2 receptor is getting down regulated, automatically patients used to have more incident with cardiovascular and respiratory system. Thereby, they are getting more accumulation of fluid in the lungs as well as hypercoagulability. So thereby, patients are dying. So that better instead will be now we are premature stage. We cannot able to give any promising statement on pharmacotherapeutic agent. As a pharmacist, we cannot able to commit also because many trials are under investigation. So that better way is maintaining our social distancing, our personal hygienicity, and also will be personal contact and also touching on the surface areas. Thereby we can prevent and also do not use same mask for for long time, and also split anything on the public places and do not contact with the living animal or it will be undercooked meat and also avoid traveling if not necessary. So main way of prevention is actually social distancing and isolation. So this way we can fight against COVID until we are having the promising agent. So far, some of the drugs showed beneficial effects, that is hydroxychloroquine, remdesivir, and avifovir, and favipiravir, and compostat. These are the agents which showed beneficial effect, but all the phase 3 trials are going on with these drugs. Unless we are having the promising agent, we cannot able to fight against COVID-19 or SARS-CoV-2. So that main thing is social distancing and personal hygienicity is very, very important. By this, I am ending my presentation. I would like to thank the management of CL Bright Mehta Institution and the Dean Madam and the Pharmacy Statistics Department, my dear friend Dr. Krishna Kumar, who taken all the pain and all the time communicating with me to deliver lecture to you all. Suggested where I have to improve my presentation. Thanks for him once again, and I would like to thank Dean Madam as well as department and institution. By this, I will hand over my presentation to the chairman for further discussion. Thank you.
Yes, thank you, Parshuraman, for your wonderful presentation. Thank you, Parshuraman, for your wonderful presentation. And now
Yeah,
the
thank
question
you.
and answer session. Our staff, our colleague, Dr. Lina, will ask you the questions. She noted the questions during the session in YouTube. Actually, the comment section, most of the audio participants they ask the questions. So now she will ask you the questions. Enlist the questions now. Dr. Lina, please welcome and ask the questions to Dr. Parshuraman. He will answer now. I think the volume is less. Increase the volume, yeah.
So the first question, does mutation make the virus more stronger? If so, how effective
actually most common with mutation is most common in bacteria, but whereas it will be in virus, RNA is not stable structure because RNA keep on will change. Here, not point of mutation, RNA keep on will change this coil structure. Same problem with HIV also. HIV also, why we are not 25 years we are not having appropriate pharmaceutical agent for virus HIV virus prevention. Same, this also will falls in the same category. Both are RNA viruses. RNA viruses, instead of going for rather for mutation, it will change this protein configuration. So mutation is not a point actually. Thing is the change of the protein structure, so that it will make the pathogen to become resistant against the drug. So the pathogen will develop resistance against the drug very rapidly compared to bacteria. Virus is develop resistant against the drug compared to bacteria. So that is mainly thing is resistant will develop against the drug.
Next question is, how long will COVID-19 going to last? Because looking back to SARS, it has given us stronger immunity for about 2 years. So how long will the immunity for COVID last?
Because this is actually premature actually. Now regarding SARS-CoV-2, we are in premature stage. We don't know immunity will work how long time. How long time will have the, because every vaccine we are having some particular time. BCG vaccine, once we taken, lifelong we will have the antibody in our body. But whereas in tetanus, the antibody will be there for only for 6 months. If vaccine come also, the vaccine, how long time that our body will have antibody against to particular virus. So that is actually under intervention. We cannot be able to promise that one because each and every vaccine they are having some particular lifetime. For example, BCG, one vaccine is enough for to produce immunity to throughout our life. But whereas tetanus, only 6 months. If any time it will be tetanus, we will go for 6 months once. Same like hepatitis B, will take for one it will be one dose and followed by booster dose for 3 times. But whereas in SARS-CoV-2, so far we are not having any clear instruction. But thing is actually passive immunity is playing some of the beneficial effects. Passive immunity means plasma transplantation, but that also we don't know how long time the host will have the passive immunity. That's also questionable.
So the next question is, can coronavirus directly attack heart muscles other than lungs?
Heart muscles, because this will be mainly smooth muscles only, because already in presentation we seen that kidney, lungs, blood vessels, not cardiac muscles, blood vessels having the angiotensin converting enzyme 2 receptors. Wherever they are having smooth muscle, especially lungs, blood vessels, kidney, they are having abundant, more amount of angiotensin converting enzyme 2 protein. So in heart, they are having the different protein. So that heart is different area, but in blood vessel will get affected. Blood vessel will get affected because blood vessel having the angiotensin 2 protein. So blood vessel, kidney and lungs are target organs for the SARS-CoV-2.
The next question, what were the reasons for withdrawal of clinical trials?
Incidents of clinical trials. But some of the trials showing that actually without giving the drugs, the mortality rate is 28%. After giving the drug like hydroxychloroquine, remdesivir, and it will be interferon, the mortality rate is reduced to 18%, thereby 10%. So the clinical trial is stating that the now current trials are not giving the complete cure but reducing the mortality. Now all the trials are there only thinking about how to reduce the mortality, because now overall if you see, some places the mortality is more than 20%, some places mortality is less than 5%. But throughout the world, they are generally if you see the mortality rate is 5%. Now everyone thinking about how to reduce mortality, because we are not having any defined pharmacotherapeutic agent for management of SARS-CoV-2, so that now all the aims going to the how to reduce, how to prevent mortality. So that also will be under interventions.
Okay. So the next question, is there any reinfection cases reported so far? So if reinfection occurs, what is the treatment?
Yeah, reported many cases. In China also, Wuhan city also reported, because initially in the China first reported cases in December 7th, then for global, January 20th they announced that China having a pandemic condition. But thing is, end of the March they stated they are free from COVID-19. But thing is actually again in April 1st week they had some of the cases. and also who were treated previously, they got reinfected with SARS-CoV-2. So indicating that the host not developing the immunity against COVID-2, SARS-CoV-2.
So is corona similar to HIV?
Not, not actually. Not. Both are different actually.
So let's just ask that participant. And we have another question. If AC inhibitors and ACE and ACE2 is used, is it different? Then why in fact hypertensive patients are affected?
Yeah, well said actually. AC inhibition, angiotensin converting enzyme, angiotensin converting enzyme 2. Angiotensin is helping to the in converting angiotensin 1 to angiotensin 2, whereas it will be directly angiotensin enzyme 2 directly acting on angiotensin 2 actually. So both are playing role on angiotensin 2. So that only when this virus is patient is getting or human is getting infected with this particular virus, it's going to down regulate angiotensin 2 system. So that will be parallelly affect angiotensin also. Even though the gene coding is different, but both are acting on angiotensin 2. Both angiotensin enzyme, angiotensin 2 enzyme, both are going to act on angiotensin 2. So thereby causing the hypercoagulability.
So how long will it take for a vaccine study?
It will take almost another 1-2 years actually, because Edward Jenner tried almost, Edward Jenner in 1880s now, we don't know what is virus also. That time we don't know what is virus also. He taken some of the things from the infected kettles and injected. From that, almost we taken 40 years to get vaccines against virus. Drug development is not a problem. Effectiveness as well as even now stem cell also under the investigation. Cancer. It will be vaccine should not produce any cancer.
So one last question. So why VHX occurs?
Immunity and it will provide agonicity.
Thank you so much for providing appropriate answers to all the questions asked by the participants. And thank you for giving your time to all of us, giving us this beautiful webinar and giving us wide knowledge on COVID-19. Thank you so much.
Thank you, madam. Thank you, chairman.
Thank you so much, Dr. Parshuraman. Your nice and wonderful presentation. This COVID-19 drug trials, drug targets, and the clinical trials. And actually, we missed one more question from our staff, our professor. What was the reason for withdrawal of clinical trials now?
Ineffectiveness and also increased number, not reducing number of mortalities. Drug ineffectiveness and also not reducing mortality when compared to the placebo groups.
Yeah, okay. And thank you for your wonderful presentation once again. And I thank all participants who all are watching this session lively. And first of all, I would like to thank our management encouraging us for all the activities our department are doing. And usually we will organize this seminar. Now webinar actually it is went well, and our department colleagues actually worked so much for this actually. I would like to mention their name, Dr. Bharati Priya, Dr. Sailaja, Dr. Leena, and Dr. Leena Upa. Actually, I organized this one along with Dr. Srilada. So we have two channels, and now I'm speaking actually. In this channel is actually I'm operating from my side, and one more channel is operating from Dr. Srilada's side. See, actually, so many times she called and we did a conversation, and after that we came with this YouTube channels, two channels we are operating now, and we will continue these two channels. And once again, I thank our principal madam who is supporting us strongly for our activities. And I thank you all participants, and my special thanks to our invited speaker, Dr. Parshuraman. So thank you all. Have a nice day. Thank you so much. Thank you.